manufacturing practices and donor eligibility for ACTPs. I'm Janet Mendonça, and I will be joined by Dr. Michael Brewer as we cover the contents of Draft Guidance Document 253, including unique considerations for complying with CGMP in the manufacture of ACTPs. All new animal drugs, including ACTPs, must be manufactured in accordance with CGMP to ensure that such drugs meet the requirement of the Federal Food, Drug, and Cosmetic Act as to safety and have the identity, strength, quality, and purity characteristics which they purport to or are represented to possess. Preserving cellular function and integrity, ensuring consistency of the process and product, and preventing contamination are critical aspects of manufacturing ACTPs, and the safety, effectiveness, and quality of an ACTP are dependent on appropriate control of the manufacturing process. There are both statutory and regulatory CGMP, and in some cases, the regulatory CGMP do not fully address those aspects of ACTP manufacturing that are necessary to ensure the safety, identity, strength, quality, and purity of the product. For example, the regulatory CGMP do not address critical items that we consider necessary to meet statutory CGMP, such as donor eligibility or recovery of ACTPs. Draft Guidance Document 253 provides recommendations for meeting statutory and applicable regulatory CGMP requirements for establishments that perform any manufacturing step in the production of an ACTP. Draft Guidance Document 253 also addresses the unique aspects of ACTP manufacturing and provides ACTP-specific examples of manufacturing practices. The recommendations in this draft guidance document are intended to help developers comply with statutory and applicable regulatory CGMP for the manufacture of ACTPs. For this part of the webinar, I will discuss establishments that manufacture ACTPs. Then I will cover recommendations to ensure quality. My colleague, Dr. Michael Brewer, will discuss the topics of tissue recovery, tissue processing, and reporting ACTP deviations to CVM. Establishments performing any part of an ACTP manufacturing process or any establishments utilized under a contract, agreement, or other arrangement for performing any step in the process must comply with statutory and applicable regulatory CGMP. If an establishment performs only part of an ACTP manufacturing process, then that establishment must comply with the aspects of CGMP that are appropriate for the manufacturing steps they perform. All ACTPs, including lower-risk ACTPs, should comply with the manufacturing standards and post-marketing responsibilities described in Guidance 218. This includes compliance with CGMP. Draft Guidance Document 253 describes two categories of ACTP manufacturing establishments, Category A and Category B. Category A establishments are those that manufacture finished ACTPs other than those finished ACTPs that pose a lower risk. Guidance 218 describes lower risk ACTPs. A lower risk ACTP is one that meets all of the following criteria. The ACTP is autologous and is intended for a homologous use in a non-food producing animal. The ACTP is minimally manipulated, and the manufacture of the ACTP does not involve the combination of the cells with another article, except for water, crystalloids, or a sterilizing, preserving, or storage agent. And the finished ACTP is not combined with or modified by the addition of any component that is a drug or device. Lower risk ACTPs are a very small group of products. Category B establishments are those that manufacture certain finished ACTPs that pose a lower risk, or those that perform steps in the manufacture of an ACTP that do not process cells or tissues for the manufacture of the finished product. An example of a Category B establishment is a unit establishment. A unit establishment is an establishment that performs a single step in the manufacturing process, such as a facility that only recovers tissue from donors. The recommendations in Draft Guidance Document 253 are applicable to both Category A and Category B establishments. Establishments manufacturing ACTPs should implement the recommendations in Draft Guidance Document 253 at the time the cells or tissues are recovered from the donor, continue through the early processing steps, and throughout the manufacturing process where appropriate, to ensure product quality and to control contamination. Category A establishments that follow the recommended ACTP-specific manufacturing practices set forth in Draft Guidance Document 253, in addition to applicable CGMP regulations in 21 CFR Parts 210 and 211, will likely be considered compliant with statutory CGMP. Regulatory CGMP for finished products should be instituted as the manufacturing process progresses. Category B establishments that follow the recommended ACTP-specific manufacturing practices set forth in Draft Guidance Document 253 will likely be considered compliant with statutory CGMP. Here are some examples to illustrate the different ACTP manufacturing establishments. In our first example, a tissue recovery facility operated by a developer collects blood from cats for mesenchymal stromal cell or MSC isolation. The same facility further processes the blood, isolates and pathogens the cells, and ultimately performs final product formulation. Note that in this scenario, the facility is performing all manufacturing steps from tissue recovery to formulation of the final product. This is an example of a Category A establishment. In our second example, a contractor collects skin biopsies from horses in a barn setting. The contractor does not perform any further processing. Instead, the contractor ships the biopsied skin to another establishment for the generation of induced pluripotent stem cells or iPSCs. This establishment that collects the skin sample is an example of a Category B establishment. Our third example is also a Category B establishment. In this example, a veterinary clinic collects skeletal muscle biopsies from dogs for myoblast isolation. The clinic washes, minces, and cools the tissue and then ships the tissue to another establishment for further processing. Note that in this example, the establishment recovering the tissue performs only initial processing steps. The tissue is shipped off to a different establishment for further processing into the finished product. This establishment that collects the muscle biopsies is an example of a Category B establishment. Next, let's discuss how establishments can maintain quality of ACTP throughout the manufacturing process. Maintaining quality is one of the most crucial aspects of ACTP manufacture and is critical for complying with CGMP. Draft Guidance Document 253 addresses many aspects important to the manufacture of quality ACTP, including topics such as establishing and maintaining a quality program, maintaining sufficient qualified and trained personnel to ensure quality manufacturing of ACTPs and compliance with CGMP, establishing and maintaining procedures appropriate to meet CGMP for all steps performed in the manufacture of the ACTP, using and maintaining facilities that are suitable to preserve quality and prevent contamination of ACTPs, employing adequate environmental controls and monitoring to prevent contamination and cross-contamination of ACTPs or equipment that may impact the quality of the ACTP. Using and maintaining equipment that is appropriate for the manufacture of the ACTP, and maintaining appropriate controls of supplies and reagents to prevent contamination and ensure ACTP quality. Due to our limited time today, I will focus my discussion on the quality program. The CGMP regulations in 21 CFR Parts 210 and 211 require establishments to maintain a quality control unit. As defined in the regulations, a quality control unit means any person or organizational element designated by the establishment to be responsible for the duties related to quality control. The quality control unit pertains to the finished product and does not address ACTP-specific considerations for preventing contamination during all manufacturing steps. A quality program, as described in Draft Guidance Document 253, is an establishment's comprehensive system for manufacturing and tracking ACTPs for all manufacturing steps performed by the establishment. Most importantly, a quality program allows flexibilities for smaller organizations, such as a unit establishment. It is not FDA's current policy to review or copy your actual quality audit reports during routine inspections. However, you should have a mechanism to demonstrate to the FDA investigator the quality audits are performed. Note that the end goal of both the quality program and quality control unit is the same: to maintain ACTP quality. The quality program is designed to prevent, detect, and correct deficiencies that may lead to circumstances that increase the risk of introduction of contamination or impact product quality. An establishment that performs any step in the manufacture of ACTPs should establish and maintain a quality program that is appropriate for the manufacturing steps performed and the specific ACTPs manufactured. This can be accomplished by defining, documenting, and implementing procedures, then following, reviewing, and as needed, revising the procedures on an ongoing basis. An establishment's quality program serves multiple functions, such as to ensure compliance with CGMP, receive, investigate, evaluate, and document information related to CGMP, take corrective action and verify that corrective actions are effective, ensure that the personnel have proper training, education, and experience, establish and maintain monitoring systems, and investigate, document, and report CGMP deviations. Here are a couple of examples that illustrate the role of a quality program in ensuring ACTP quality. Our first example relates to the cat example, where a Category A establishment collects blood from feline donors for the manufacture of an MSC product. Now let's discuss a scenario where the recovered blood is suspected to have bacterial contamination. What should the establishment do? The establishment's quality program should already have established procedures for assessing risk and determining appropriate follow-up steps in the event of a possible contamination of the ACTP. There are many possible actions that should be taken by the quality program to address this situation. Two examples of actions that the establishment should take include: conduct an investigation to determine if any suspected tissue has been distributed and initiate recall procedures if necessary, and to assess their tissue collection SOPs for any deficiencies. Note that these are just two examples of actions that should be taken, and additional actions may be necessary. Now let's discuss a scenario for a Category B establishment. In our horse example, a contractor collects skin biopsies from horses and then ships the biopsied skin to another establishment for generation of iPSCs. What happens if the facility that is receiving the biopsied skin tissue finds out that the shipping containers are not properly sealed? What should the receiving establishment do? The quality program should ensure that appropriate corrective actions relating to manufacturing practices are taken and documented, and verify that such corrective actions are effective in accordance with CGMP. There are many possible actions that should be taken by the quality program to address this situation. Two examples of actions that should be taken include the facility receiving the shipping of the biopsied skin should review product quality testing to determine if tissue is acceptable for further manufacturing. Also, the establishment shipping the skin tissue should re-qualify the shipping container materials. I will now hand it over to Dr. Michael Brewer.
Thank you, Dr. Mendoza. Hello, everyone. I am Dr. Michael Brewer, and I am a chemistry, manufacturing, and controls reviewer on the biotherapeutics team in the Center for Veterinary Medicine, Office of New Animal Drug Evaluation, Division of Manufacturing Technologies. I'm going to continue now with a discussion of unique aspects of ACTPs that should be evaluated and controlled for the purpose of quality manufacturing, starting with tissue recovery. Due to the limited time available during this webinar, I'm going to focus on recovery and then continue with processing and processing controls, as well as process changes and validation. Please do refer to Draft Guidance Document 253 for further information and recommendations for labeling, storage, and receipt, pre-distribution, and distribution. All of these sections of Draft Guidance Document 253 are critical to ensuring ACTP quality for both Category A and B establishments. As stated in Draft Guidance Document 253, recovery means obtaining from a donor cells or tissues intended for use in implantation, transplantation, infusion, or transfer into an animal recipient. This can include minimally invasive blood draws or skin biopsies, as well as more invasive surgical resection of adipose tissue or bone. Controlling the recovery process is one of the first steps to ensuring quality manufacturing. The establishment that performs the recovery should have validated procedures and records. This is expected whether the establishment is solely contracted for recovery procedures and then ships the material for further processing, or if the establishment performs recovery procedures and then conducts further aspects of product processing through final formulation. Procedures at recovery establishments should consider the risks of contamination and cross-contamination that may occur. Risks are often associated with the procedures, personnel, equipment and supplies, or the environment of the recovery facility. Additionally, a tracking system that follows every batch and dose of the finished ACTP should be traceable back to the initial tissue recovery process. This ensures traceability from donor to recipient should any quality concerns arise or if disease transmission is suspected. If tissue recovery occurs at a contract establishment, the developer is still responsible for ensuring that the controls are adequate and that all involved persons are compliant with CGMP. Let us consider several examples of how procedures and records should be implemented to be compliant with CGMP. In the equine IPSC example, the receiving establishment observed at receipt that the shipping containers were not sealed properly. This deviation has the potential to adversely impact the quality of the shipped material. As just two examples of actions that should be taken to meet CGMP, the receiving establishment should have a procedure for placing the incoming material in quarantine. Additionally, all findings should be documented in the batch records, including the results of testing which is conducted to assess the material's quality. Further, procedures that are followed in response to the deviation and any corrective and preventative actions that are implemented should also be documented. All of these steps may involve coordination by the developer for both the tissue recovery and tissue processing establishments. Additional actions may be necessary to meet CGMP. In the canine myoblast example, contamination in the harvested tissue is suspected to have been introduced by a technician that handled the tissue. As just two examples of actions that should be taken to meet CGMP, The establishment should implement quarantine procedures to isolate the suspected tissue. The establishment should also review training of the technician to determine if they have been appropriately trained for the function they are assigned. Just like the equine example, the initial deviation, procedures that are followed in response to the deviation, and any corrective and preventative actions that are implemented should be documented. Additional actions may be necessary to meet CGMP. Note that both deviation examples involve different types of establishment scenarios. The equine example involves both a Category A and a Category B establishment, whereas the canine example only involves a Category B establishment. However, both of the described scenarios are capable of meeting CGMP for ACTPs. We will now consider several examples of how contamination should be controlled to be compliant with CGMP. Let's first discuss the feline MSC isolation example, where blood collection is completed in a tissue recovery establishment. This establishment should ensure that personnel involved in the recovery operations have the necessary education, experience, and training. Personnel should also have their qualifications and training reviewed periodically to ensure they can perform their roles competently. In addition to assessing the personnel qualifications, establishments should assess their recovery equipment and validate their cleaning and sanitization processes to prevent the introduction of contamination. Cleaning programs should be supported by environmental monitoring where appropriate, and each establishment should assess the appropriate frequency, method, and concentration of disinfectants used to ensure prevention of contamination and cross-contamination. These are just two examples of actions that should be taken, and additional actions may be necessary to meet CGMP. In comparison, the barn in which the equine skin biopsies are collected is not expected to have certain equipment, such as HEPA filters or even an air handling unit. However, this establishment should assess the recovery procedures and evaluate the impact of the air quality on the collected tissue. Controls should be implemented to prevent contamination, such as restricting tissue recovery to a location away from the dust of a riding ring, or only permitting recovery at a time when other matter is not actively displaced into the air, as could occur during feeding. While the recovery area of the barn may not be capable of a similar level of sterility assurance relative to the tissue recovery facility in the feline example, the barn establishment should still develop cleanliness and handling procedures that can be demonstrated to prevent contamination. Again, these are just two examples of actions that should be taken, and additional actions may be necessary to meet CGMP. I'm going to continue now with a discussion of how tissue processing should be evaluated and controlled for the purpose of quality ACTP manufacturing. To discuss tissue processing, I'm going to focus on processing and processing controls, as well as process changes and validation. As stated in Draft Guidance Document 253, processing is defined as any activity performed on an ACTP other than recovery, donor screening, donor testing, storage, labeling, packaging, or distribution. In particular, processing can include microorganism testing, preparation, sterilization, adventitious agent removal or inactivation, and procedures associated with storage, such as preservation and removal. Sterilization processing may be associated with the manufacture of ACTPs that do not have a mechanism of action which is dependent on the activity of live cells or tissues. Developers should establish controls throughout the manufacturing process. This should include the development and execution of mechanisms to monitor manufacturing processes and in-process testing to assess for contamination. Every process that is performed should be considered for the potential risk of introducing manufacturing variability and contamination or cross-contamination. Developers should also establish controls and testing for the recovered cells and tissues, which can mitigate risk associated with the raw material. It is important for this discussion on tissue processing to include a few notes about pooling. Pooling can involve the combination of starting material collected from a single donor from several independent recovery events. Additionally, pooling can involve the combination of starting material collected from multiple donors. In general, CVM does not support the practice of pooling because pooling presents an increased risk of cross-contamination and disease transmission from donor to recipient. However, if you determine that pooling of ACTPs is necessary, for example, to obtain a therapeutic dose, we recommend that you discuss this with CVM. For developers with questions and concerns about their proposed product development, CVM highly recommends conversations early in the development process. We encourage you to take advantage of our Veterinary Innovation Program for this purpose. Let's look now at a few examples related to processing and controls. In the canine myoblast example, skeletal muscle biopsies undergo processing such as washing, mincing, cooling, and myoblast isolation to manufacture the final product. To meet CGMP, the developer should develop a standard operating procedure, or SOP, that details these and all processing steps required to produce the myoblast product. SOPs are often implemented and documented as a batch record and should be strictly followed during the manufacturing process. Developers should also establish a tracking system that can distinguish between different donors and batches. To meet CGMP, in the canine myoblast example, a myoblast dose should be traceable to both the batch associated with the dose and to the canine donor from which the skeletal muscle was originally collected. The tracking system should also permit traceability between the multiple establishments associated with product manufacturing. Developers should also ensure that the material has been adequately evaluated for contamination risk. Considering the feline blood-derived MSC example, to meet CGMP, the developer should ensure that the tests used are adequately sensitive and specific to detect contamination of the collected blood, which presents a risk for disease transmission from donor to recipient. Further, the in-process testing that is performed should be validated to ensure that decisions made during manufacturing are based on accurate and precise data. Developers can establish a decision tree that dictates whether the next step of a manufacturing process, such as cell culture harvesting, can begin based on the results of in-process testing. Now we will discuss two examples related to changes in validation. As an example of a proposed change to the manufacturing process, developers may want to consider alternative means of manufacturing, filling, or storage. In the equine iPSC example, the developer wants to change the tissue storage temperature from -80 degrees Celsius to -20 degrees Celsius to reduce costs and improve ease of transport. To meet CGMP and the recommendations in draft guidance document 253, the developer should conduct real-time stability studies to collect data in support of this change. Developers should also consider all critical quality attributes that could be impacted at the alternative storage temperature and ensure that the analytical methods utilized are stability indicating. Manufacturing processes can change as additional data are collected or unexpected events occur. In the canine myoblast example, the developer has been using fetal bovine serum, or FBS, from a company that goes out of business. In this situation, the developer will need to qualify an alternate supplier and fully evaluate whether the new FBS reagent will impact their manufacturing process. These actions are important because FBS is of animal origin and inherently brings a risk for introducing contamination into the manufacturing process. FBS is also a reagent that is susceptible to compositional variability, which can have significant impact on the growth of cells or tissue in culture. Therefore, to meet CGMP recommendations related to aspects of tissue processing, developers should adequately qualify their vendors as well as fully evaluate if the reagents will impact their manufacturing process and quality of the marketed product. The final portion of this presentation on draft guidance document 253 will discuss CGMP deviations and reporting to the FDA. Due to the limited time available during this webinar, I will not be able to discuss CVM's recommendations for records, tracking, and complaint files, which are the remaining topic areas described in draft guidance document 253. Please refer to draft guidance document 253 for further information and recommendations for these topic areas. However, please note that both records and tracking have been briefly addressed when previously discussing examples associated with procedures and records and processing and controls. CGMP deviations are deviations from the applicable sections in Draft Guidance Document 253 or from applicable standards or established specifications that relate to preserving quality or preventing contamination of the ACTP. All CGMP deviations should be documented. When an establishment identifies a CGMP deviation that may result in adverse events, product defects, or manufacturing defects, the establishment should report the CGMP deviation to the developer and to other establishments that may be impacted by the deviation. These may include establishments performing downstream processing steps on the same ACTP, establishments using tissue from the same donor, and establishments and developers of other ACTPs that you process that may be impacted by the deviation. The developer should report CGMP deviations to the FDA within 3 working days if the ACTP is in distribution and the CGMP deviation may result in an adverse event. For approved ACTPs, you must also comply with the reporting requirements in Title 21 of the Code of Federal Regulations, Part 514.80 for adverse events and product defects. In the first example shown on the screen, after distributing a batch of an approved feline MSC product, the establishment manufacturing the product identifies that retained samples for this batch of product are out of specification for acceptable impurity limits. The batch contains elevated levels of endotoxin, which may result in serious adverse events if administered to recipient cats. As this product defect may result in a serious adverse event, the sponsor must submit a report to the FDA field office within 3 working days. For more information on how to submit these, please consult our Adverse Event Reporting for Manufacturers website. If the sponsor receives reports of adverse events, those adverse events are also reported to the FDA separately. In the second example pictured on the screen, the establishment that performs the initial processing steps for the equine iPSC product detected contamination with mycoplasma. The cells were already transferred to a second establishment that manufactures the finished iPSC product. However, the product was not yet distributed. The initial establishment should follow their protocols upon detection of contamination and documentation of the investigation, and any follow-up actions should be initiated. As the batch was not released for distribution, there is no potential for adverse events. In this case, a 3-day field alert report is not submitted to the FDA. However, because the deviation may result in adverse events or product or manufacturing defects, the initial establishment should submit a report of the CGMP deviation to the developer and to the second manufacturing establishment. Both establishments should maintain documentation of the CGMP deviation and make this information available upon request, such as during an on-site CGMP inspection. This assessment can be utilized by the FDA to determine if manufacturing facilities and developers appropriately detect, investigate, and implement corrective and preventative actions. The manufacture of ACTPs is complex and introduces considerations that may be atypical for traditional pharmaceuticals. CVM recognizes that the regulatory CGMP described in Title 21 of the Code of Federal Regulations, Parts 210 and 211, do not specifically or fully address all aspects of ACTP manufacturing and may not always be applicable to the unique nature of ACTPs. Therefore, Draft Guidance Document 253 provides CVM's recommendations for meeting both statutory and applicable regulatory CGMP when manufacturing ACTPs. The recommendations in this draft guidance document should be applied to consistently produce quality ACTPs and to ensure that ACTPs are not contaminated and do not become contaminated during manufacturing. To further promote this process, CVM invites developers to discuss your product and its unique characteristics with us early in its development. We also recommend enrolling in our veterinary innovation program to facilitate communication regarding manufacturing practices and CGMP compliance. Dr. Janet Mendoza and I have discussed some of CBM's recommendations for CGMP for ACTPs as described in Draft Guidance Document 253. In the third video of the webinar, Dr. Jan Linkenhoker will discuss Draft Guidance Document 254 and donor eligibility for ACTPs.